Hi, everyone. So first of all, welcome. Thank you very much for coming to this symposium, trying to find a solution or method that we can employ to combat this reproducibility crisis. And I just want to thank you, Dr. Freeman and Dr. Edwards, for taking the time to come out and share with us their thoughts on what can be done to this problem that is looming in research. And I also want to thank H2I for helping us arrange this facility so that we can host everyone here to this talk today. So once again, my name is Tom. I'm one of the founder of BenchSci. We take a little bit different approach than the previous talks on how to validate antibodies. We believe that the vendor do play a very important role in solving this issue, but they just don't have the power or the money to test every single one of the antibody with every single use case that's possible. So we rely on the literature that scientists are good and they publish good data, and we try to build a method to get this data to you when you decide on a reagent to purchase for your experiment. So I'm just going to start with a story. Why should I care? I was talking to some of my colleagues about coming to this talk, and some of the response that I get is, why do I need to care about reproducing experiments? As long as I'm good, I publish my research, that's great. I can publish my paper, continue my thesis, so on and so forth. But I can tell you a story when I was doing my PhD that one of my friends sent me this paper from one of the very high journal, and with a very interesting protocol that would actually propel my research a few months forward. So I was very excited, and then I take this paper, I read it, I understand it, I buy all the reagents, and then I test it. After 1 or 2 times, nothing works, so I was thinking, yeah, probably my mistake, I'm not diligent enough. But after months of trying, then I started to get very frustrated. And then I call up the author of this paper and ask, hey, can you give me a more detailed protocol? And then they send me something which is a little bit different from what they have in the paper. And then I realized the reason why I cannot reproduce the data, because on their Western blot, they just name the complex name of the protein they're targeting and not the subunits. So if they were be able to tell me that subunit that you're looking at is actually not going to be detected and cite the antibody citation, then I would be able to understand a lot sooner that what I'm trying to look at is actually not detected in their Western blot. And that just shows you how important it is for people to cite things properly so that scientists around the world can properly reproduce your data so they can get on with their thesis, get on with their project. As a matter of fact, as we have alluded before, not just that people are not citing, but even if out of the antibodies out there, 50% of them do not work. So if you have a new target today, you buy 6 random antibodies from a company, chances are half of them is not going to work and you've wasted your time and money. And the funding situation, Canada, US, is always stringent. We really don't have the capacity to waste this time and waste this funding. And the paper showed before, $28 billion every year is on preclinical research that is not repeatable. And a major culprit of this is on the antibody. Why is it so difficult to choose a good antibody? I think the reason is mainly twofold. The first is that there are just so many of these antibodies out there. Companies just keep on making and making and making, and they do not spend the energy to test them. So based on what we have, our own number shows that there are roughly 3.7, nowadays maybe even higher than this number, and supplied by 150 vendors. So choosing from these antibodies is just not possible. And our number also show that for the more popular protein, roughly there are 3,000 to 5,000 different catalog numbers that you can choose from. So it's really kind of like by luck that you can find something that works. Compounded by the fact that 50% of them do not work, good luck looking for a good antibody for your experiment. But this is not all that bad, because there are data out there that is useful for helping you choose antibody products. And as I've mentioned before, these data are hidden in publications. So there are good scientists out there doing good experiments, showing that something worked before. It's very simple, right? If someone else have done this experiment, you look at the data looks reliable and correct to you, chances are if you buy the same antibody, it will also work for your experiment also. But this also brings out another problem. Databases nowadays are not best suited for looking into the pieces in the materials and methods. For example, PubMed or Google Scholar, chances are you will find something like antibody inhibitor X, block mutation Y on protein Z. This is probably quite easy to look for them. But going into the materials and method, they just not designed to do that. So if you try PCNA or PCN antibody on Google, you will get tens or maybe tens of thousands of results. And as a BC scientist such as yourself here, you really don't have time to go in and find out which of these results carry the data that you want to look at. And then this creates a huge problem. On the one hand, you have many antibodies to choose from. Some of them are good, some of them will help you with your thesis, make you publish that paper, but 50% of them not work. So choosing one is difficult. On the other hand, you have lots of paper. These papers contain information that you want to digest in order to help you choose, and scientists is stuck in the middle. Both sides are very difficult to navigate, very difficult to find things. And this is definitely slowing down biomedical discovery. Consider these scenarios. So based on my experience and talking to scientists, usually we buy around 3 to 6 antibodies to choose one that works for our experiment. We validate them, we spend time, knock out cell line, knockdowns, and takes hundreds and up to thousands of dollars maybe, and take days to weeks to validate this antibody in-house. And if you so unfortunate picking a wrong antibody, the cost will be higher now because your Western blot, all the cells that you've been cultivated, the gels and the time that you've spent, now you will have wasted thousands of dollars maybe in reagent costs and also weeks to months wasted. But that is nothing compared to using the wrong antibody and not knowing about it. You think that it's working, you don't know that it's binding something that you don't want to bind or for other reasons, then the direct cost is going to be hundreds of thousands of dollars. And I didn't make this number up, right? Because from the paper, the Nature paper shows that a very prominent researcher in Mount Sinai Hospital wasted thousands of hundreds of dollars and months of research because something that he thought binding to the target is not actually binding at all. And that would definitely waste you months or years and your research, and the other lab will have published that paper in Nature, and there goes your Nature publication. So here at BenchSci, our mission is to try to solve this problem by helping you find the best antibody. And by the best antibody, we mean to bring you to the product that has the most validation in the paper. And then you can decide whether this validation or this information provided in the paper is suitable for your experimental context. So basically what we did is we built a computer to read like scientists can. Human scientists are great in critical thinking, thinking about hypotheses, testing experiments. We want to have a computer to do the more mundane and tedious task, to look for data in the paper that shows you if a reagent works. I think scientists should be doing something whereas the machine can do these other things. And to build this kind of algorithm, we combine a lot of expertise from many different areas, such as bioinformaticists, computational biologists, cell biologists, and definitely machine learning experts. So what we do is, for the first step that we've done is we mapped proteins to the proteome. All the antibodies that we have is mapped to some kind of pathway from open data sources. This is very important because as scientists, we all know that we write things many different ways. We all have our own name of calling our favorite protein. We might know a few of them, but we never know every single one name of our favorite gene. And if you go onto the publication now to search for things, chances are you will not get the complete picture. But by mapping every single protein or antibody product to the proteome, we will employ a search one, search all strategy, so they give you a more complete picture of all the data that is out there, so you don't have to look for every single name and drop down those papers that associate with it. Furthermore, we don't just want to understand if something is cited. As there's been alluded before, things might be cited, but it might not be useful for your experiment. If you go to some other organization, they tell you, hey, antibody X is cited 600 times. therefore it's good. Actually, it's not. I remember the story when I was doing my PhD. I go to my PI because my PI is very stringent on finding only good antibodies to use. And I would tell him, hey, this antibody is used 600 times in so many publications. It must be good, so can I buy this for my experiment? And then he would just laugh at me and say, Tom, have you read this paper? Did you understand in this paper if this product is good for you or not? If not, then you cannot go buy this product. So knowing if something is cited is not useful. And as a matter of fact, for something that is maybe produced 10, 15 years ago, of course they will have accumulated a certain number of publications. But what is to say that this other newer antibody that is released a month ago, 2 years ago, is not as good as this one, right? So to understand the context and the use case around the antibody is very important. That is why we do not just understand if an antibody is cited, we also want to understand the biology behind the citation, how it is used in the paper and where it is used. And we want to bring this information to you to make a more educated guess or decision when you want to buy the product. We take all these things that we collect from the paper, the image, the text, and the citation now, and then we build a machine learning algorithm around it. What we did is we have a team of 10 PhD scientists labeling a lot of figures, labeling sentences as how a human would interpret this data. And then we build a algorithm around it, and the outcome is that we will have a use case for you. So you're choosing an antibody product for ERK1, PCNA, whatever, and we tell you where it has been used and how it is used. The very interesting fact is that it takes a team of 10 PhD scientists over a year to label 1 million sentences for the computer. The computer can read 4 million sentences overnight, just like that. So because we have an algorithm to read the sentences and the figures, we can bring you the most relevant data for your research. If you pick these 4 random search terms, for example, PCNA in human, immunohistochemistry, and liver, and you look at it in PubMed, you will find over 6,000 papers to read because they do a keyword search. It's not targeted and it's not specified. But on our platform, with our technology, we will bring you only 13 specific figures. And this is a lot more relevant to your research because you know that this data is surrounding the search term that you're entering. So instead of wasting time reading paper, reading other people's work on reagent that might not work, read these ones and decide which antibody is useful for your research. And the other important thing is the algorithm that we designed can read all the literature out there. So instead of going to one publication, seeing this one very beautiful Western blot, and decide to buy this product, we tell you the history of this product because we're able to go back many years to build a more comprehensive picture of how it is used. Oh, this is an antibody used in Western blot, and out of the 500 citations, 499 of them is used for Western blot. Then you must know that this antibody is pretty good for Western blot. Versus the other one, maybe it's one beautiful figure, but everything else is used in chip or other technique, then you will think that, this might be just a one-off negative experiment results. So what we have built is we're trying to transform the antibody selection process to make it a lot more easy and faster. So before, you might need to test 3 to 6 antibodies, and you might be using something that is not correct for your context, not the same cell line, not the same technique. But with BenchSci, with this technology, you only need to test 1 to 2 because you know that the things that you're buying is supported by literature results. And you will be able to find something that is used in similar situations. So whereas this one is used in 293, I don't know if it works in HeLa, we will show you that yes, this antibody has indeed worked in your tissue of interest, your cell of interest. And we transform the not knowing into knowing. So it makes you have more confidence in something that will work for experiments so that you won't waste time and money testing things out. And that's what I want to present about the platform. We're going to move in to give you a quick product demo, and then I will take questions afterwards. So let me switch to this here. Okay, so this is going to be, let me see, yes. So this is open to academics. Any one of you can go back and make an account and then speed up your research a lot faster. Believe me, I talk to a lot of scientists. I show this to them, and they say, boy, I just hoped that I have this when I was doing my research, because this is a problem that's close to myself. So Basically, I'm building a tool for myself if I were 5 years ago. So we can start as a cloud-based platform, so you can come onto benchsci.com from anywhere. You can do it on a bus if you want. You can do it in your lab, anywhere with internet connection. So let's start off with choosing a protein that I was always using when I was doing my research. PCNA is a replication fork protein. So after you type in your query, what is shown right directly underneath are search terms that are most relevant or most similar to what you enter. But what is more interesting are things that are shown below it. So you can see in the bracket the source antigen or the organism that is used to raise the antibody. So whether it is a zebrafish PCNA, mouse PCNA, human PCNA, and what have you. So let's select the human PCNA. And as I've mentioned before, you see a list of aliases. We help you choose and select the other, look for the other aliases as well. So what you see immediately in this page are publication figures. So we're very different from other companies out there that tells you about publication, because we don't just tell you this one? Yeah. So we don't just give you a list of PMID so you can go back home and print the paper to read. What we show you here are figures that we collected directly from those publications. So you can ingest and understand the data right away instead of reading through the abstract, looking for the skew, looking for the materials and methods. So these figures here all contain a commercially available antibody that target human PCNA. And also we remove the frustration of reading the paper, seeing this beautiful figure, only to find that this antibody is a gift from the other lab. Please refer to my publication 5 years ago to find the catalog number. So we don't have any of this because we have the citation in the paper. So this is where you can find the published figures, but we also partner with the vendors in order to obtain the images and to populate it here. So some people may say, okay, vendor images, I'm skeptical around it, but for some other people, this is the minimum requirement for them to decide if an antibody actually worked at all. So we also analyze these images with our technology for the image and for the context around it. And then also, there's nothing for this protein yet, but we are encouraging students, encouraging postdocs to submit their own images, because we all know we buy 10, we buy 6, we publish the best one. The other ones we probably tested. I have like gigs of data in my hard disk with controls that I have no use, but I have them lying around. So we are encouraging scientists to submit this more raw, untouched Photoshop image to help the other scientists to decide whether this antibody is a good one. And lastly, we also partner with some, so to speak, third-party or independent validation organization, such as the Human Protein Atlas or the ENCODE database, to have them put their images on here, their own validation of different antibody products. So what we're trying to do essentially here is that we want to put in front of you all sources of validation or experimental results possible so that you can make a better choice in selecting your reagent. So let's get back to the figure from publication for a second here. So you can see we have around 1,200 images that we have collected, and you will be thinking, there's so many, how am I going to go through these images to find my product? So what we have on the side, therefore, is a list of filters. So because our technology read the paper beforehand, we can tell you exactly what happened in this figure. So a very popular filter would be, okay, I'm doing a Western blot, I'm doing a immunofluorescence, what have you. Then you can choose the technique that you want to do. Then immediately you can see that the figures now change to a panel of very pretty IC images. And then another popular filter would be company filter. There will be companies that you like, for example, CST. I quite like CST because they have very robust criteria before they release a product. But there are also some things that you might not like. So this is a place where you can select something to include or select something to exclude. Also, we have some other filters here, such as host, reactivity, clonality, so basic filters for you to decide what kind of product you want to look at. What is also something new that we've added is the specificity of the antibody. let's say you are doing a cloning experiment and I cloned in the C-terminal domain of this protein. I need an antibody that will target the C-terminal domain, not some other region. So you can choose the specificity of your antibody here. So let's say C-terminal domain. And then you can see now the figures continue to filter down, getting closer and closer to what you want to do. And then we have some other filters here. For example, the PTM filter is useful if you're looking for a specific phosphoserine, tyrosine serine type antibody, or a methyl acetyl type protein, or tissue. I'm doing experiment in liver cells, liver tissue, brain tissue, cell lines. So let's say today we want to do something in the liver. So now you can see from that 1,000 images, we boil down to just this one. If you go to PubMed, you will find thousands of papers, and then not all of them is relevant to your search. But here, because we read it for you, we find you this one that satisfies all your 4 search criteria. IHC image, that has to be C-terminal domain of a human pCNA antibody. So now you can conveniently click into the image and see the figure itself. So we also have a very cool magnifying glass function here. So you can look at the image in more detail. So you can see this is an IHC experiment with pCNA in liver tissue. And the legend is on the side. So for you, quick reference of what happened in the figure, what is red, what is green, so on and so forth. And then the publication itself is also situated here for you to see which paper is published from. Then if you want to read more about the paper, the full text is just a click away for you to look at the abstracts, look at the discussion, look at more materials and method if you so choose. So the product that is used in this figure is provided underneath here. So you can see this is an Abcam product, anti-pCNA, and in here it is being performed with IHC experiment. So this is what I was mentioning before. Because we read everything, we tell you the history of this product. So not only would you understand in this situation it is used for immunostaining and IHC, you will also know that it is being used for Western blot in some other situation and also immunofluorescence. So you can know what is the best application for this antibody. You can click into this product page to learn more about this antibody. The history bar that I was mentioning is a little bit bigger in the product page, so you can see what other techniques that it has been used for. What we've added recently by talking to scientists is the drilling down a little bit on the technique now. So not just we tell you it's IHC, we tell you that it's been used for formalin fixed tissue and also paraffin embedded tissue. So that you know what kind of experimental context is being applied here. And then the antibody information is on the side. All the data that this antibody has ever generated is on the bottom here for you to kind of see how good this antibody is in other situations. And then you can apply the filter again. Oh, let's see how this guy do with Western blot. Then you can filter this antibody, filter these figures down to see how this works for Western blot. And then some other things are on the side here. What are the cell lines it's been used for? What are the cell type? So on and so forth. And then in the end, you can click the vendor page link out, and then we will bring you to the vendor page. One important thing to mention here is that we do not receive any kind of compensation from the company. So there's no, this antibody is better than the other one because they pay us $5 to say so kind of thing. No advertisement. Anything you see here is ranked or sorted by algorithm by most recent publication. So you can know that if we say that this is the publication, it's not because the vendor wants us to say so, but it's because it is legit data we collect from publication. So this is one very interesting way to use BenchSci where you want to do a little exploration yourself. You want to go read the paper, read the figure, and then see what is produced from the figure. But some other scientists are more in a rush. I just want to know what is out there, out of whatever you offer or whatever the community offer that can satisfy my need right now. So we also have a question, answer for that one. So let's say we choose the same protein again here. So instead of reading the figures, you can switch over to the product tab. So for PCNA human, we know that there are around 3,000 antibodies out there that target this protein. That is made possible because our bioinformaticists link all the antibodies to the proteomics. And they are sorted by how many times it's cited, so you can see immediately what is more popular in the literature, so to speak, and what is not. But the cool thing is that you can apply the same filter as we did before. So let's say Western blot, and let's say in liver tissue. Then you can find out that from the 3,000 antibodies, these 14 satisfy what you are looking for, and it has been published in the paper. So instead of throwing a dart on a wall and find out, oh, which of these 3,000 would probably works for Western blot and liver, we tell you these ones work before in the literature for liver and Western blot. And then you can click in for one of them to further read the data that they produced, and then you can find out the clonality. This is a PC10 monoclonal antibody from Abcam, and then you can go to the vendor page and purchase it. Or even choose a few if you want to test a couple to make sure you have find one that would work for you. And then lastly, something else that I want to show is a kind of a side use of BenchSci. So of course this is great for you to look for reagent. Right now we're doing antibody. Very soon we're moving into plasmids, sRNA, shRNA. Anything that hits a protein, we will be moving into those reagents. But another use is to kind of do a more catch up on the current literature. Because paper comes out every day. Like in PubMed, 1 million papers added every year. So for you as a diligent scientist to kind of catch up on all this new data is just not very possible if you want to have a life. So what you can do is you can start your search with not a protein, with some other tissue that you're interested in. For example, oh, I'm starting a new project, I'm working on glial cells. I want to know what kind of biomarkers are great for this tissue or cell type. So you can start off by typing in glial, and then on the image side, you will see images that are relevant to the tissue type or whatever context that you enter. The cool thing is, if you switch over to the product side, you will be able to find out these antibodies is most used for this tissue type. Then you will know, oh, NUN is highly used as a good biomarker for glial, or GFAP is also another good marker for glial cells. And then now you can say, I am planning a IF experiments on glial cells. What kind of antibodies is useful for that? And then we can tell you this antibody from EMD Millipore produced a lot of IF results with the glial tissue type. And then you can go to the side and you can do a quick browse on all the nice images produced with this antibody in glial tissue. So with the combination of the filter, the possibility is endless. Whatever you want to see, oh, I wonder if someone did this, I wonder if someone did that. Instead of telling you a paper, a link to read the paper, we tell you exactly how and where the experiment is performed and the data that associate with it. So you don't need to read text that is maybe not relevant for what you're looking, because looking at the figure is always the most satisfying thing. And then also maybe you have performed a Western blot, and then oh, the protein is 100 kilodalton, but my band is 80. I wonder what's going on. Is this something wrong with the antibody? Is this something wrong with what I've done? You can come here and type in the product and then go Western blot and see what other have been done in Western blot with your protein of interest and see if that data actually agree with each other. So this is a more kind of discovery path of using BenchSci. So that's all I have for you guys today. We'll be happy to take any questions if you have.